The genetic code is like life's instruction manual, guiding how every living thing grows and works. Let's look at its main features and how this shared language connects all living organisms. The genetic code is set of rules used by living cells to translate information encoded in DNA or RNA sequences into proteins. This process is essential for life as proteins are the building blocks and the functional unit of cells. The genetic code is read in triplets of nucleotides which are known as codons. The four nucleotides in RNA are adenine, which is denoted by A, uracil denoted by U, cytosine denoted by C, and guanine denoted by G. In case of DNA, instead of uracil there is thymine, which is denoted by T. They combine in groups of 3 to form codons, each of which corresponds to one of the 20 standard amino acids required for protein synthesis. With 64 possible codons, it is clear that there are more codons than amino acids, which introduce us to the concept of degeneracy in the genetic code. The concept of genetic code was postulated by George Gamow, who proposed the idea of triplet code, setting the foundation for our current understanding of how genetic information is translated. Now let's see how to read the genetic code table. Locate the mRNA codon. The mRNA codon made up of 3 RNA bases. Since RNA uses uracil instead of thymine, codon will consist of the bases A, U, C or G. By using the table, we will find the first base of the codon along the left side of the table, and the second base along the top of the table, and the third base on the table's right side. Where the row and the column intersect, we will find the amino acid that corresponds to that codon. For example, if we try to locate codon CCU in the genetic code table, we have to find cytosine or C in the leftmost column for the first position of the codon. Next, we have to locate C or cytosine along the top row for the second position of the codon. Finally, find U or uracil in the rightmost column for the third position of the codon. The intersection of these will form the codon. The genetic code table is the reference for the relationship between codons and in mRNA and the amino acid they specify during protein synthesis. There are total 64 possible codons. Out of 64, 61 codon specify amino acid and rest 3 codon codes for stop signals, which are stop codons. Now let's talk about the key features of the genetic code. The genetic code is read in sets of 3 nucleotides and are called codons, and they are made up of 3 nucleotides which codes for amino acid. For example, the codon AUG codes for amino acid methionine, which is also the start codon. This triplet nature allows for 64 possible combinations, which is sufficient to code all amino acid. The genetic code is nearly universal across all living organisms, meaning that the same codon code for the same amino acid in nearly every species. The codon UUU codes for phenylalanine in humans, bacteria, and even plants. This universality is a key piece of evidence for common evolutionary origin of life. Specificity or unambiguous. Each codon specifies only one amino acid, meaning the genetic code is unambiguous. For example, the codon AAA will always code for amino acid lysine, and it will never code for anything else. Degeneracy, or the genetic code is degenerate, meaning that multiple codon can code for the same amino acid. For example, the amino acid serine, which is encoded by 6 different codons, and these are UCU, UCC, UCA, UCG, AGU, and AGC. This redundancy provides a buffer against mutation. Non-overlapping nature. The genetic code is non-overlapping, meaning that each nucleotide in a codon is part of only one codon. This ensures that the reading frame is maintained during translation, and mutation typically affects only one amino acid in a protein. For example, in the mRNA sequence, AUGCCGAUU, the codons are AUG, CCG, and AUU. The genetic code is comma-less, meaning there are no punctuation marks like commas between codons. The reading frame is continuous and each codon is read one after another without any interruption. This allows for a smooth and continuous translation of genetic sequence into a protein. Chain initiation codons, also known as start codons. The translation process begins with the specific codon known as the start codon. The most common start codon is AUG, which codes for the amino acid methionine. This signals the start of the protein synthesis and sets the reading frame. AUG codes for methionine in eukaryotes and formyl methionine in the prokaryotes. Chain termination codon or stop codon, and also known as nonsense codon. Stop codon signals the end of the protein synthesis. These codons do not correspond to any amino acid but instead indicate that the translation process should stop. The three stop codons are UAG, which is also known as amber, UGA, also known as opal, and UAA, also known as orche codon. When the ribosome encounters one of these codons, the protein chain is released. And that's how the genetic codes unlock the secret of life. That's all for this video. Thank you.